So thank you very much, first of all, to the organizers for this fantastic initiative. I'm really looking forward to the next 2 days and hearing about all the bioarchaeological research that's going on across different regions of the Roman world. And yes, so I'm going to focus today on Crete, but then also set that within the context of Greece. Because basically, whenever I started to put this together, I realized just how little has been done on human skeletal remains on Crete. You know, this is a common theme, I suspect, that's going to appear. So I'm, want to look at Greece more widely as well. And, even for Roman Greece, there's relatively little still has been done. And I'm defining that by the modern borders, of course, because that's what we have to deal with when it comes to applying for permission from the local archaeological services. And we also have to set the limits somewhere for what we're looking at. But of course, these are meaningless borders in terms of the Roman world. This region encompassed, 4 different Roman provinces. We've got Macedonia, Epirus, Achaia, and then Crete. But there's also the Aegean Islands, whose provincial status is a little less known. And we have no Roman bioarchaeological studies coming from there yet. But this would be a very nice thing to change in the future. This is a very interesting and dynamic region. So yes, I'm firstly going to look at what's been done to date on Roman bioarchaeological studies in Crete, with a particular focus on the project that I've done at Knossos. But I'll also talk briefly about new work that I started this summer at Aptera, and then I'm going to broaden it out to the rest of Greece, and then look at the future directions that we need to be going in. So the personnel that we've got working on Roman Crete at the moment, Chryssia Bourbu, who works at the local Effort of Antiquities for West Crete in Hania. And she mostly works on Byzantine material, is interested in the bioarchaeology of infants and children, and is now with the new archaeological museum that's opened in Hania. She's been working a lot on the sort of ethics of displaying human remains in museums. We've got me in the middle there. And then there's also Susan Kirkpatrick-Smith, who works at Kennesaw State University. And her work's not limited to Crete or the Roman period, but she's been running an osteological field school, which I'll say a little bit more about in a minute. So until very recently, this was the only publication of any human skeletal remains from Crete. This paper, which looked at 2 sites, which Hellenistic Hania and then Roman Sphakaki. Looking at biological and social status, it interpreted the individuals in these assemblages as elite due to the types of tomb construction and the rich grave goods, and looked at a variety of paleopathology, essentially. But the value of this study really lies in its attempt to compare 2 populations. And because this sort of comparative study was completely lacking, especially for these time periods in Greece. That's not without problems. We're looking at different time periods at different sites, so it's hard to talk about change over time. And such studies are still rare, largely due to the lack of well-excavated cemeteries covering multiple time periods. Most excavations of cemetery contexts in Crete and in Greece are rescue excavations. And I was very lucky for my PhD project to have access to an assemblage that gave good coverage across approximately 1,000 years at a single site. And I'll say more about that as well in a minute. So this is the Greek osteology research project, which Susan Kirkpatrick-Smith has been running since 2007. And it's looked at human remains from a variety of sites and time periods, and is based at the INSTAP Study Center for East Crete. But the human remains that they're looking at the moment come from a Roman cemetery of Erapitra. And this is really an interesting cemetery with very nice Roman built tombs. So I really hope we will see results of this work published in the future. And I've also been talking about collaborating with this project to try and do some biodistance work in the future. So if we broaden out the temporal boundaries of what we call Roman, there's been a lot of study done on what's really a Proto-Byzantine population from Eleftheria on Crete. And this is work again by Chryssia Bourbu. And I've just highlighted, this is this is hopefully all of the bibliography that I could find, highlighted some of the key words. So what she's been looking at, we've got paleopathology, health. She's done carbon and nitrogen isotopes for dietary reconstruction, and also looking at breastfeeding and weaning patterns. And then if we stretch our temporal boundaries back in the other direction, looking at the Hellenistic period, we also have a very small number of studies on Crete for that. So 2 studies, again by Bourbu, from Hanya, one looking at a Hellenistic mass burial, and the other looking at the association between dis and higher status individuals. And then there's my work at Knossos, which was Hellenistic, Roman, and late antique. And I'll talk a little bit more about my project. So it focused on health, diet, and life ways. So looking at paleopathology, stable carbon and nitrogen isotope analysis, and looking at the importance of prevalences. So as the late, great Tony Waldron always drummed into his students, we've got to look at all of these things from a paleoepidemiological perspective. So understanding the occurrence of disease within a population or a group-wide setting, and not just observing incidences. So I looked at change over time throughout these time periods, but also looked at intra-population variation by age and sex, and also by social status as inferred by the tomb architecture. And of course, there are problems with inferring social status from the burial type, which we won't get into now, but we'll just proceed with the necessary caution and caveats that comes with that. And the methods I used focused within the areas of demography. So, I looked at age at death and sex, worked out a minimum number of individuals, I looked at activity, so within that joint disease and enthesial changes, diet, and then growth and development during childhood. So the Knossos assemblage, we all like to have a little complain about the condition of our material and always assume that everyone else has better preservation within their material. But my material came from excavations from back to the 1930s through to the 1980s, and these were excavations of the British School at Athens. And it was a mixture of both individual and communal graves. So the Hellenistic assemblage was largely individual graves, and the Roman and late antique largely communal. So therefore, a lot of the material was very commingled. It was fairly highly fragmentary. And because these are old excavations, they suffered from the usual problems of storage and preservation, which sort of impacts the commingling problem sometimes. And I conducted my recording by individual where that was possible, but by element or partial element if necessary in an access database in order to record regardless of preservation and work out an MNI basically. So I ended up studying the material from 109 tombs with a minimum number of 537 individuals, 232 of which were Roman. And in this case, I'm defining Roman as from the end of the invasion of Crete, which was in 67 BCE, through to approximately 400 CE. And we have adults and sub-adults, males and females, but the sub-adults do appear to be underrepresented in all time periods. And this was a particular problem for the Hellenistic assemblage. We definitely had remarkably few sub-adults, and where they did appear in the assemblage, they were buried with adults. So there were definitely some other burial practices going on for the sub-adults in this time period. So it's hard to compare a full age at death profile due to the lack of juvenile burials for the Hellenistic period. But if we compare just the adult age at death profiles, although there's no significant difference between any of these, and you can see the difference for the young adult group marked out with the error bars there, the overlap that shows that there's no significant difference. But there is a slightly larger proportion of young adult deaths in the Hellenistic period, and there are more people living into old adulthood in the Roman period. So I also looked at change in diet over time from both the dental conditions and stable isotope analysis. And we see an increase in caries, calculus, and antemortem tooth loss from the Hellenistic to Roman period, and a decrease again in both caries and antemortem tooth loss in the late antique period, though the calculus does go up quite significantly. But I suggest that this may mean more sugars in the diet in the Roman period compared to the Hellenistic, due to sort of increased production during this time period, and increased connectivity and trade networks. So more products being produced on Crete, but also new foodstuffs potentially coming in within these networks. The stable isotopes, again, didn't show very significant differences in diet. There are a few individuals at all time periods who appear to have had a small marine component in their diets. But the mean carbon values from Hellenistic, so the Roman period, did increase slightly. So there's a suggestion that possibly something like millet, a C4 plant, was coming in as a subsidiary crop in the Roman period. So looking at social inequality as well. So I've just picked out a couple of examples of things I looked at. So looking at the prevalences of caries and osteoarthritis for individuals in different tomb types. And some slight trends can be seen, though the sample sizes were small for some of the tomb types. So the tombs are ordered from most elaborate to the simplest grave forms. And what we can see is higher caries in the built and chamber tombs than in the pit and tile graves. We see the lowest osteoarthritis in the built tombs, so the most elaborate tombs. But again, so this is depicting all time periods together here. So the osteotheques were all late antique. The pit graves were largely Hellenistic. So there's the results may be slightly skewed by change over time as well here. So I'm looking to expand my research out to look at more sites on Crete. So far, I made a start at Aptera this summer. So the Aptera study focused on a 2010 excavation by Katerina Zanakaki, who works for the Effort of Antiquities of Hanya. And the plan is to look at differential connectivity at different sites in the Roman period at this time of intensified connectivity for Crete, and look at the differential impact on health and lifeways. So more to come on that, hopefully. So spreading our gaze out to look at Greece more broadly, the earliest studies of human remains in Greece were some of the earliest studies in bioarchaeology or osteoarchaeology as a discipline, and these varied from very traditional approaches with not doing much besides measuring skulls, to what was quite pioneering work in the field at the time by the likes of Lawrence Angel, who was seeking to understand the causes behind pathological lesions that we were observing in the bones. But it wasn't until the late 90s or early 2000s that we see people like Sherry Fox and Anna Laya, who were actually using osteological approaches to understand aspects of life in the past of Roman Greece, with their studies at Corinth and Athens, respectively. And since then, the numbers of studies have increased, though slowly. And as you can see from the bibliography here, there are not vast numbers of studies. I've mentioned a couple of studies here that are still in works in progress. There have been a couple of PhD theses on human remains at Corinth. But even there, where more study has been done, there's never been a study that really brings everything together, all these disparate studies, and looks at the bigger picture, as well as integrating it within the archaeological and historical context. So although there's a lot more that can be done, we've got lots of legacy assemblages and recent excavations with material, but there has been a lot of progress as well made in recent years with several new and ongoing studies. Some of these PhD projects by the likes of Frisa Vergidou and Anna Carli-Ghiotti and Vola Trizzaroli's postdoctoral project at Dion. And we see new methods being incorporated into these studies. There are several dietary isotope studies that have been published both by myself at Knossos, by Anna Lagia for Athens and Lavrion, and several others which will be published in the very near future. And we also have the first biodistance study for Roman Greece by Effie Nikita and colleagues at Akfraia in Boeotia in central Greece. But Roman Greece in general has been a slightly neglected topic in the archaeological field in general. Roman Crete in particular is definitely vastly understudied, but at the same time it's a quickly growing field and it's receiving more attention. We've had several conferences, conference sessions, publications come out in recent years, but books like these, osteoarchaeological studies, are completely lacking. So there's a lack of synthesis in these studies. But regardless of the shortcomings of these volumes, the absence of osteoarchaeological study highlights firstly that it's still an understudied discipline, but also it needs to be integrated into the bigger history, the social history narrative. And there's so much that we can get from our field, and that archaeological and historical narratives would benefit from our work for looking at this contextualized picture. So we need more interdisciplinarity, and I'm aware I'm running out of time, so I'll sort of round things up quickly. We need to think about putting archaeology at the core of these studies, having an integrated approach to mortuary archaeology, ensuring that we have a mainstream role. We need to make our data accessible. We need to make legacy material accessible by collaborating with local authorities, collaborating with new excavation projects, talking between different specialisms. I think one of the best ways to get things done is with PhD projects. Funding is always a problem. Here at the University of Groningen, we do have this collaborative PhD funding where we will fund 2 years of a PhD project if another institution will match that with another 2 years. So there are always possibilities and there's lots to be studied. And yes, thank you for listening. And our Groningen Human Osteoarchaeology Lab is a very new and growing group. We started our Instagram page yesterday, so you can follow us there. But yes, thank you very much.